Good afternoon, everybody. I'm Vince Kinsler, the President and Executive Director of the Parkinson Association of Greater Daytona Beach, and welcome to our interactive webinar series, You, Me, and PD, with today's program titled The Genetics of PD, Progress and Implications. She is a friend, an advocate, and a tireless worker for people with Parkinson's in the greater Central and North Florida communities. We're just so pleased to welcome our dear friend, Dr. Alaine Keebaugh. She'll be talking to us about the genetics of PD. So join me in welcoming Dr. Keebaugh.
All right, well, thank you, everyone. Thanks, Vince, Christina, and Marianne for having me. I want to thank you all for joining us today. As we're all well aware, these are difficult times in our country, and we're thankful for you all to take the time to connect with us today. And so I want to give you all just a little bit of background on myself. So many of you know me, I'm Alaine Keebaugh, and I spent the first decade of my career studying human genetics at Emory University in Atlanta, Georgia. And I was interested in studying genetics because I've always been fascinated by how humans can be so similar and yet so different at the same time. And I thought that the answer must be in our genes. And during my research tenure, we did some really cool things like alter gene expression in the brain so we could study things like love and addiction. We deleted genes and then we put genes back in so we could study movement disorders like tremor and dystonias. And we even developed novel ways to create new genomes from birth, and we were able to turn these animals green when it worked. And it was, while this science was really cool, I also began to realize that it doesn't really matter how much we learn if we learn it in a vacuum. And so 6 years ago, I transitioned out of academic research and into industry. And one of my passions is to share what we have learned in the lab so that real people can be impacted by this science. And my goal today is to share with all of you what I know about the genetics of Parkinson's disease in a way that we can all understand. And there's a lot of really cool research going on out there, and it's going to begin to change the way that Parkinson's is treated here in the near future, and hopefully one day it will lead to a cure. So I'm going to talk a little bit about Parkinson's disease, but I'm not going to spend a whole lot of time on this. And that's mostly in part because you all probably know more about Parkinson's disease than I do. You know, you guys live with it and you manage it on a daily basis. But for those of you who may not be as familiar and joined us, Parkinson's is a progressive degenerative movement disorder. It's marked by a decrease in dopamine producing cells in the brain. The exact cause of this disease remains unknown. It's pretty prevalent. It affects 1 million Americans and more than 10 million people worldwide. And for those of us who are lucky enough to live over the age of 55, 1% of us will be affected by a diagnosis in our lifetime. Now, Parkinson's is most well known for its motor symptoms, and these symptoms can make everyday life challenging. They include things like rigidity, tremor, shuffling of steps, stiffness, and slowed movement. And even though Parkinson's is most well known for these motor symptoms, the non-motor symptoms can oftentimes have just as big of an impact on the quality of life, and many of them may even present before a diagnosis. And so these include things like loss of smell, changes in mood, pain, insomnia, and fatigue. So in most people, when we talk about their Parkinson's disease, we say that it is idiopathic, which means we don't know what caused it. And that's actually the case for the vast majority of people with Parkinson's disease. But in a handful of people with PD, 15% is our estimate, we actually do know what caused the disease. And in these people, something has gone wrong in at least one of their genes. And what we're going to do today is we're going to start with a review of genetics, and we're going to use that as our foundation for understanding what this means, not only for people with a genetic form of Parkinson's disease, but for anyone with Parkinson's disease. And we're going to focus on words that a lot of you have probably heard before, and you may or may not know what they mean. And these are going to be things like DNA, gene, protein, mutation, genetic variation, different forms of inheritance, penetrance. And my goal for today is that by the end of today's talk, everyone will know what these words mean, and they will understand how they relate to Parkinson's disease and why it's important no matter what your Parkinson's disease is, whether it's idiopathic or genetic. Okay, so let's start with DNA. So what is DNA? So DNA is the deoxyribonucleic acid. It is found in almost every cell of your body, and it provides the template for making a human being. It is the hereditary material that's in all humans and almost all other organisms on Earth. And what's interesting too is that nearly every single cell in your body has the same DNA. And so what is this DNA made up of? So it's made up of chemical bases. They're right here. It's called adenine, guanine, cytosine, and thymine. And if you look over here to the right, you'll see that adenine and thymine bind together and cytosine and guanine bind together. And when they bind together, they hold this DNA backbone together. And if you look at this DNA as it's stretched out, it kind of looks like a ladder, and it's a ladder that's twisted. So it's a lot of times it's referred to as a spiral or a double helix, and it's packaged really tightly. So it's wrapped around these proteins that are called histones, and the histones are wrapped around each other. And then eventually it condenses into a structure known as a chromosome. And we'll talk a little bit more about that in a second. Okay. So we have our DNA, and then we have a gene. So what is a gene? A gene is actually just a part of the DNA. It's a sequence. It's a small section of the DNA. So you can see this gene here. And it gives instructions for building a protein. And this is the protein here. So this is our gene that's found along the stretch of DNA. These are the nucleotides, so the adenine, guanine, cytosine, and thymine that are in a particular order. And that order, of course, is different for each gene. And this is basically the instructions that are given to make a protein. And proteins are the workers of the cell. They basically do everything that you can think of. They perform functions like providing structure to the cell, transporting molecules, they fight infections, they get rid of waste, and they carry out various chemical reactions. Humans have approximately 20,000 genes, right? And these genes, like we said, are made up of a sequence. And here in this first picture, we have what we call a normal sequence, or geneticists might refer to this as a wild type sequence. And we can have mutations that arise in this sequence. So here we might have a mutant where a guanine is changed to an adenine. And I think, one of the most important things to realize about mutations is that they are not all bad. Sometimes they actually have no significance at all. So they can go from having no effect to causing disease, or to just causing subtle differences between people. And I think that's something that's really important to remember as we talk about the genetics of Parkinson's disease. Okay, so where is a gene found? So a gene is found on a chromosome in a specific location called a locus. So here you see the chromosome, and anywhere along this you can have a gene, and there's nomenclature for that, but I think that's probably beyond the scope of this. So it's important to remember that the chromosomes are tightly packaged, and each chromosome contains thousands of genes. And these chromosomes are then put into the nucleus of a cell. And every single cell in your body has the same chromosomes, the same DNA. So one thing we should talk about is, where do these chromosomes come from? So you probably all have heard in some form or another that everyone has 2 copies of each chromosome. So we get 1 copy from our mom, and we get another copy from our dad. A female has 2 X chromosomes, so she's always going to pass on 1 copy of an X, and a male has an X and a Y, and so he's either going to pass on the X or the Y. And this is why it's often said that the dad determines the sex of the baby, because the mom always contributes an X while the dad contributes either an X or a Y. Let's go back to where I said that we can have mutations, and they may vary in significance from having no significance to impacting disease. So let's talk a little bit more about that. So what that's called is genetic variation, and genetic variation is the difference in genes between individuals within a population. So let's look at this wild type gene here. So this is an example of a gene sequence. And this is what the average person in the population might have. And then let's say, for example, we take someone with Parkinson's disease and we sequence their genes, and we want to see if we can identify a difference that might be causing their Parkinson's disease. So let's say down here we find a mutation, and that mutation is where the guanine has been changed to an adenine. So here, the way that's labeled is the guanine would be changed to an adenine, and this would take place at base pair site 6055. So I just wanted everyone to kind of see how a mutation is labeled, so that if you see that in the literature or you happen to be, reading about something, you understand what they mean by that. And so one thing to point out about this is, let's say that this mutation was identified in someone with Parkinson's disease. It's important to remember that this doesn't necessarily mean that this particular mutation is actually causing the disease or has any relevance in it. And so that's one of the things we should talk about. What happens when a researcher or a physician identifies a mutation, and what does it mean? How do they go about figuring out if it's causal or not? And so to understand this, I want to talk about how genes are inherited. Okay. We're going to talk about two types of inheritance. So one type is autosomal dominant, and that's right here. So this kind of is a cartoon image of chromosomes that you say inherited from your mom and from your dad. So in the autosomal dominant case, you only need to inherit one copy of that mutation to have the disease or the causal implication, whereas with autosomal recessive, you would need to have two copies. So you would inherit one copy from the mom and one copy from your dad. Now, our early understanding of genetics was that one gene causes one disease. And we now know today that it's much more complex than this. And so I mention this because I want to, say from the beginning that from here on out, when we talk about a gene mutation that causes disease, we can either mean that it definitely causes the disease or that it will increase your chance of causing the disease. And these two things are a little different. And as we go on, I think you'll understand why it's so important to point this out. But, let's say this mutation down here is actually a mutation in the LRRK2 gene, and we're going to talk more about that gene here in a minute. But in this gene alone, there have been identified more than a handful of different mutations, and each one of those mutations can have a different outcome into whether or not it causes the disease, or it just increases your chance of the disease, or it has no impact at all. And so I think it's important to remember that while 5 of us may all have a mutation in the LRRK2 gene, our mutations might actually be different, so it could mean different things for each of us. So let's talk a little bit about autosomal recessive and what that inheritance pattern really looks like. So I'm a person that I like to have things, see them visually to understand. So we're going to talk about an example of autosomal recessive. So in this case, the father and the mother both carry a gene that is known to be associated with, let's say, Parkinson's disease. And that's represented here by this little b. So the big B would be the wild type. The little b would be the copy that carries the mutation. And so what does this mean for their offspring? So neither parent has the disease, right? Because it's autosomal recessive, they would need two copies. But what about their children? So the way that geneticists figure this out is you put one of the parent's genotype at the top of this square here, and the square is what we call a Punnett square. And you put the other parent's genotype across the other side, and then you mate them. This is what the genotypes of the offspring would look like. So one possibility would be the offspring could have a big B and big B, means they don't carry the mutation. There's also a 50% chance that the offspring could have a big B and a little b, which means they look just like their parents. So they carry a copy of the gene, but not enough to actually influence the disease. And then there is a 25% chance that one of their offspring could carry both copies of the gene. And so you can imagine how in an autosomal recessive scenario, genetic variation is maintained in the population, right? Because you can carry this genetic variation without it having any significance on your disease outcome. Now let's look at, and please, if you guys have questions, or Vince, if you want to hop in
Yes,
and,
I do have a question there, Elaine.
okay.
Talk about both copies of that gene and then a 25% chance, I think, of receiving both copies. Is that what that 25% represents?
That's right. It's kind of like the chance that if both a mom and a dad who carry that mutation were to have a child, the child would have a 25% chance of carrying that both copies of that mutation.
Thank you. Earlier, you'd mentioned that, these components, the dynamics, if you will, of, here, let me go. The dynamics of people carrying these genes doesn't necessarily mean that they're going to get Parkinson's. So, but you put it into a couple of different categories. Definitely causes, increases the possibility of something happen, and then possibly of no impact. If somebody in this characterization is in the BB, the small b category, the 25% carrying on, does that put them at some greater potential? Of what potential and what category does that put them?
Yeah, that's a great question. And I wish I had, a very clear black or white answer, but I don't. So it depends on what that mutation is and what the mutation causes. So let's say that this little b mutation causes the LARC protein to not even be made, then yes, that person would have the disease. Most cases, these mutations don't always penetrate through. So you could be a carrier with a little b and still not get the disease, but you would have, say, an increased risk somewhere between 20 and 80%. And the reason for that, Vince, probably is that some of these mutations probably just give you are kind of like a genetic risk factor, and maybe there's like an environmental insult that has to happen, and these two things have to happen together to cause the disease to
That,
come out.
that actually kind of makes sense the way you explained it, because from what I have learned over the years about Parkinson's, I've known people where it was pervasive in families, and yet certain people in those families it just didn't happen to, and yet they know that they had the predisposition genetically to have the disease, and yet never got it. So it just, I think, lends itself to the dynamics of this syndrome.
Yeah.
It's difficult to hone in on these things.
It's hard, yeah, and on the same line where you might have the genetic predisposition and then you have an environmental insult that sort of tips you over, the same could be true, and we don't know, is maybe you have the genetic predisposition and then there's something about your lifestyle choices that protects you. Say, probably not smoking, but let's go with exercise,
How about
something like
coffee?
that. And coffee,
How
yes.
about coffee? Well, and I think that's where we're going with that. Why certain people do get it and why certain people don't get it. Anyways, amazing. Thank you.
Yep.
Good.
Good. Okay, so we're going to go on to autosomal dominant, and Vince, please chime in when you have questions. So
Sure.
the next type of inheritance pattern is the autosomal dominant form. And if you look here, you'll see the father has a big B, little b. And again, in this case, the little b is going to denote the mutation. But the difference is you only need one copy to have the disease. So in this case, the dad has we'll say Parkinson's disease, and the mother's unaffected. And so what are the chances that their offspring are going to also carry this mutation? So we'll set our Punnett square up with the father's genotype here across the top, the mother's genotype across the side. And when we cross these, you see that there's a 50% chance that the offspring will carry both big Bs and they won't have an increased risk for the disease. And then there's also a 50% chance that the offspring would have a copy of the mutated gene and be at an increased risk for the disease. And so I hope that what you guys can see is that depending on what type of inheritance pattern you have, autosomal recessive or autosomal dominant, it can influence how pervasive it is, especially within families, and how sometimes it might be hidden between generations before we actually see it. And I want to talk a little bit about this before we move on and talk more specifically about what these genes are, is to touch on heritable genetic variation, which is what this is and what we're talking about, and that it's not always a bad thing, right? And it's what makes people different. It's what makes animals different. It is this heritable genetic variation that is what evolution actually acts on and allows us to evolve and change with our environment. And so an example of that, we're going to talk about tiger coat color, mainly because tigers are my favorite animal. So if we look here, you'll see there's a tiger with a red coat and a tiger with a white coat. And let's say they fall in love and they have offspring. And their offspring are going to get a copy of the white coat gene from their mom and a copy of the red coat gene from their dad. And so what these offspring look like are, they all have an R and a W. And so in this case, that would make them have an orange coat color. And what's interesting here is that in this case, none of the offspring actually look like the parents. But if we take it out another generation and we say, okay, now we have two orange tigers that mate and have offspring, what are their offspring going to look like? And we fill out our Punnett square. And now you see that we could have one offspring with a red coat, two offsprings with orange coats. So there'd be a 50% chance that they look like their parents, and then one offspring with a white coat. And we're actually back to where we started. And so in this way, genetic variation can be maintained in a population. And then sometimes selection can act upon it. Let's say that these tigers somehow now live in the Arctic, or up on a glacier, and the white coat actually blends in pretty well, and so they can't be predated or hunted on, and they might actually be more successful. And in that way, the white coat gene might be selected upon and become more prevalent in the population. So it's just an example. I hope that made sense to you. And so what I'm trying to get across to everyone here is that our genes have a large influence on our lives. They affect what we look like, they affect our personalities, our preferences, and our susceptibility to disease. And by studying genetic variation, researchers hope to better understand the cause of human disease, ultimately to be able to better predict disease outcomes, and then to be able to treat disease and ultimately cure it. And so that's kind of what the second half of this talk is going to focus on, is how can we harness this genetic information to influence better outcome, better treatment choices, and ultimately maybe even a cure. Okay, so let's talk about genes that we know are associated with Parkinson's disease. So currently we know of 23 regions of DNA that contain mutations associated with Parkinson's disease. And some of these gene regions are associated with known genes, and some of them aren't. They're just kind of in these spaces of genetic material. We don't really know what gene is there. And then on top of that, there can also be multiple variations within a single gene, so different types of mutations that can infer a risk of developing Parkinson's disease. So let's take a look at this chart here. So if you look over here, you'll see the genes listed, and the first one is SNCA. We've got Parkin. Here is one that is unknown. And they also have different inheritance patterns. So this SNCA is dominant, so it's autosomal dominant. So we know that you only need one copy in that case, and it confers a risk factor. So that doesn't mean that you're going to get the disease, but it means you now have an increased risk of getting the disease. If you look at Parkin, it's a recessive gene, and you can go on down the list and you see that it's all different kinds. And if you look at GABA or GBA here, it can be autosomal dominant, autosomal recessive, a risk factor. So there's lots of different variations here, and it can get kind of confusing. If you look here, it's to tell you the location on the chromosome, which we didn't really talk about. And over here you have variants. So if we look at just the SNCA gene, you can see to date there are 5 point mutations that have been identified. There's whole gene multiplications, deletions. If you look here in the Parkin gene, there's over 250 different mutations. And so this really kind of drives home the point of, just because you have a mutation in that gene, it doesn't mean that you're definitely going to get the disease or even have an increased risk. We kind of have to go in and interrogate these different mutations to understand what they do. All right. So here's a list of autosomal recessive genes. So we have Parkin, PINK1, FBXO7, ATP13A2, and DJ1, just to name a few, and I'm going to use these as an example. So as these genes were discovered, investigators tried to study where they were expressed in the cell, where they were expressed in the body, and what they wanted to do was see if they could find some way that they were all connected. Um, but what they ended up finding was that the proteins from these genes were expressed in different cellular compartments. They were expressed in different organs in the body. And so there really didn't seem to be a very clear link between what these proteins were doing and how these mutations were playing a role in Parkinson's disease. But scientists continued at it, and what they eventually came to realize was that these proteins were actually all linked in circuits. And so by studying all of these different genes and these mutations, they were able to identify a canonical pathway for which neurons die. And so what we've learned is that all of these genetic events in this diagram actually lead to the inactivation of Parkin. And this ultimately results in the activation of a nuclear surveillance protein that's called PARP1, and that's in the pathway right here. And generally, we refer to PARP1 as the guardian of the genome. It keeps everything in check. But when this gene sees too much DNA damage or too much cellular injury, it ends up activating the cellular death cascade. And so what we've learned is that all of these really rare genetic events have led us to better understand how neurons die. And this is not just the case for Parkinson's disease, but it's also for other neurological diseases as well. And the reason that this is important, the reason that we should care about this, whether we have genetic forms of Parkinson's or not, is that this opens up new avenues to develop new drugs and therapies for everyone with Parkinson's disease, as well as it bleeds over into other neurological disorders as well. And so let's talk a little bit about some of the autosomal dominant genes and what role they play in the pathways.
I had a
And I'm going to
question about
stop
that,
for a second. Yep.
Elaine.
What?
When they identify some of those genetic events that you were referring to, for instance, when we speak in terms of the medications that are constantly coming out to help treat Parkinson's or the symptoms of Parkinson's, what do they do? I mean, how do they even, there's so many different medications and they're developed differently. For instance, we know certain diabetes or cancer-related medications are now used to be treating symptoms of Parkinson's. How do they know that? How does somebody that's researching Parkinson's, do they take that neuron path and they put it on a petri dish and start injecting different chemicals? How do they come up with these formulas to treat these symptoms that way?
Well, that's a really good question. And I have an example of that at the end of this talk. And it's the idea of we can actually repurpose old drugs for new therapies, right? So I'll jump ahead a little bit and just kind of set the stage for that. So
That's
within,
just one component. That's, old drugs treating, current different symptoms of different
yes.
diseases. I'm talking about, how about the development, the molecules of the medicines they're creating, like the carbidopa, levodopa. Was that an existing medication that they found out helped with Parkinson's, or was it developed specifically for Parkinson's? And if it was, how in the heck did they know?
So
I'm
I can't
curious. It's
answer
interesting.
that question specifically because I'm not a pharmacologist, but so with carbidopa levodopa, right, it's the gold standard that we go to, and the reason they tried it is because they know that carbidopa levodopa is a synthetic form of dopamine, right? And we know that in Parkinson's disease, the dopaminergic neurons die. And so that's that drug is based off knowing the biology of Parkinson's disease at the very basic level. So if you look at this diagram here, so we have Parkin, PINK1, FBXO7, and after I go through some of the dominant forms, we're going to look at this pathway again and talk about where might researchers identify places in this pathway that they could develop drugs, right, to help go after Parkinson. So one might be that they have a PARS inhibitor or a PARP1 inhibitor, could have a PINK1 activator. And so the short answer to your question is that by understanding these pathways, they can then go in and look at these pathways and see if there are drugs that could inhibit where the pathway goes wrong. So that mutation leads to some alternative function or gets rid of a function that we need, then they can develop drugs to target that. And yes, first they would test it in a petri dish, right? And then they would move on to, say, an animal model in mice and primates. And then if those results look good, they would move into humans.
It's amazing. Thank
It
you.
is.
Really cool.
All right, and I'm going to talk more about that in a minute. So if that answer did not satisfy you, we'll go more. And I have a question here. Can I get a copy of the Park gene regions? My dad has Parkinson's, and now my sister and I both have the disease. Yeah, I will send out a list of those genes to Vince, and he can distribute it to the people who have joined today. We're going to move now and talk a little bit about the autosomal dominant forms of the disease. And you all probably heard a lot about alpha-synuclein, and one is because when this protein is misfolded, it does lead to the disease. It was also the first genetic cause of Parkinson's that was identified back in 1997. So within this gene, there are many different point mutations that can facilitate the misfolding of alpha-synuclein. It can also be duplicated and triplicated. And so when this happens, it's not necessarily that the protein is misfolding or altered in some way, but you have a lot more of it available, which presents more opportunity for it to accumulate and misfold and ultimately become pathogenic. This gene was first described in 1997, and again, it said it was the first one. And since then, it's been extensively studied. So I found it fascinating that if you were to do a PubMed search, would be looking at peer-reviewed articles on this topic. From 1997 to today, you would find over 8,500 publications on just this gene alone. So that means while we don't know everything we need to know, a lot of people are looking into this. Another example of an autosomal dominant is VSP35. And this is a protein that it sits in the protein synthesis and sorting machinery. And the reason that's important is because we know what it does. We were able to find it. What's interesting is that when it's not functioning properly, you get alpha-synuclein accumulation, you get loss of dopamine neurons, and you get a Parkinsonian-like phenotype. Researchers have also identified that it interacts with another protein that is of an autosomal dominant form of PD, LRRK2, and this in turn triggers the activation of the AIMP pathway, which triggers the cell death cascade that's activated by Parkin that we talked about in the autosomal recessive pathway. So you guys should be starting to hear that some things are starting to sound a lot alike, right? Whether it's autosomal recessive, autosomal dominant. And so again, by studying these genetic forms, even though they're really rare, we're starting to learn even more about how dopamine neurons die. I do want to spend a little bit more time than I did on the last two talking about LRRK2, and that's because there has been a lot of work around this gene, but more notably, the founder of Google has these mutations, and so he's done a lot of work advocating for research into this gene. He's very interested in understanding how these mutations cause Parkinson's disease, but also how this information might be used to treat Parkinson's patients. So this gene is the most common cause of genetic forms of Parkinson's disease. I believe about 5% of people will have a LRRK2 mutation. It is both familial in origin, meaning that it's inherited from the parental line, but also sporadic, meaning that during your lifetime, you can accumulate mutations in this gene that you maybe not were born with. It's a big protein, and there are mutation sites that are clustered into different functional domains of the protein. So if you look down here, this is think of it as a cartoon of what this protein looks like. But the mutations that are associated with Parkinson's disease are clustered. So they're clustered here, and they're clustered here, and then there's one down here. And if you look at, these are genetic sequences down here from different other species. So here we have Gallus gallus, which is a chicken. We have, let's see, the rat, the mouse, Homo sapiens, that's humans, cow, C. elegans, which is a worm. And all this is really telling us is that this protein or this gene is really highly conserved from humans all the way back to C. elegans, which is an earthworm. And so the fact that we see mutations here probably means that if a mutation were to arise here, it would be deadly, right? So we wouldn't survive with that mutation. So this is an old gene. It's found across many species of organisms, and it seems to be doing a lot of very important things. And so, like all genes that are linked to Parkinson's disease, this particular one is widely expressed throughout the body. So it's expressed in the brain, the lung, heart, kidney, gut, muscle, skin, and blood. And so where it's expressed in the body doesn't really seem to give us any clues about how it might be contributing to Parkinson's disease. There are mutations in this gene that are also linked to things like cancer and Crohn's disease. And so if we want to figure out how to inhibit LRRK2 function, this presents a bit of a challenge, right? Because it's expressed outside of the nervous system and it's doing a lot of very important things in some very important organs. We also are beginning to learn that it plays an important role in the immune system, both in immune cells in the brain, which would be these guys here, the astrocytes, oligodendrites, and the activated microglia, but also plays an important role in the peripheral blood and immune system outside of the brain. And so, you know, when you have LRRK2 that's not functional, you also have issues with autoimmune disorders like Crohn's disease and inflammatory bowel syndrome. So it's really a big challenge here to figure out how to regulate LRRK2 in Parkinson's without causing additional problems. And so that's kind of one of the things that we should talk a little bit about. So what does a researcher do once they identify a new gene that's been mutated and they want to figure out why it causes disease? Researchers will do this type of analysis where they want to figure out what the day jobs are, and they want to separate those from the dirty jobs. Dirty jobs would be what's causing the disease, while day jobs are things that it does every day that don't contribute to disease. And a lot of time is spent trying to sort through these. And they look for, maybe they can identify problems with protein synthesis or abnormal protein expression, or, you know, problems at the synapse that might prevent communication between two neurons. And with regards to LRRK2, they have actually been able to implicate that there are issues in RNA translation. So RNA translation would be when you go from it's an in-between step between the DNA, the gene sequence, and the protein. So the gene's not actually getting to make the protein the right way. And they've also found that this can also be a mediator of neurodegeneration, which makes sense from the pathways that we talked about a minute ago. And so, so this work is still in its infancy, but by beginning to be able to understand these things, we will be able to identify new therapeutics and ultimately block the development of Parkinson's disease. So let's go back to our pathway, right? So this was the pathway that I pulled up before. It's just arranged a little bit different. So this is, we also have, there should be a second part to this. There we go. So up here we have the autosomal dominant pathway, and you can see all these different things have been identified, and it leads to the cell death cascade. And then we also have the autosomal recessive. And what understanding the biology of this, so all these rare genetic events that researchers have been able to study has led to the identification of potential therapeutics in this pathway. So Vince, this is getting at the question that you asked. So we have identified a potential therapeutic that can be used here, one that can be used here, ABL inhibitors, PARP inhibitors, MIF inhibitors, and Paris inhibitors. And so these are all things now, these are all new novel therapeutics that researchers can go after to try and help either treat Parkinson's disease, maybe slow the onset of it, or prevent it from showing up at all. And so, you know, a lot of times, you know, you hear people maybe of the mindset where if it's this rare genetic thing, why do I care? It doesn't impact me. Why are scientists spending so much money and time trying to understand this? And the reason for that is a couple things. One, that genetics is allowing for a better understanding of the biology of Parkinson's disease. And by having this better understanding of the biology, we're able to identify and develop new therapies. And hopefully at some point in the very near future, this will usher in a new era of personalized medicine, right? And the next slide, we're going to talk about two examples. We're going to talk about what's in the pipeline for two genes, GBA and LRRK2. And before we go there, though, I do want to just remind everyone that, up until very recently, even people with extensive family histories of Parkinson's disease did not receive genetic testing. And the reason for this is because there were no clear use for the test results, right? So there's so much variability in clinical characteristics in the mutations themselves, even with people who have the same mutation, that there's really no clear practical implication for knowing your genetic status. So, it doesn't impact how your Parkinson's disease is treated or managed. And so other than just, contributing your data to science, there wasn't a big value for that. But that's beginning to change. While there are no approved drugs yet based off genetics, there are clinical trials. So let's talk a little bit about what's out there and what's coming. So as we said, there's currently no genetic therapies that are available and that are approved. But knowing your genetic makeup can be informative because it can allow you to participate in some of the genetic studies. That does not mean that you should all go out and join these genetic studies, because there are pros and cons to that as well. But let's talk about what's out there and what's coming. So we know that mutations in LRRK2 increase activity of this protein. And so researchers have developed therapies to reduce its activity. And specific, we're going to talk about Denali Therapeutics. They have found a drug that they've proven is safe, and it's well tolerated in healthy volunteers. They found that in the fluid surrounding the brain, these study participants had high levels of the drug. So this suggests that the drug is going where it needs to go. And the next step now is for them to test this drug in people with Parkinson's. And this is actually underway. And it should be pretty far along, and we should be hearing about these study results, I would expect, this year. So probably close to the end of the year. And Denali Therapeutics isn't the only company developing drugs to target LRRK2. So for the 1% to 2% of people with a LRRK2 mutation, other companies are looking at drugs that can potentially slow the progression of the disease. But not only for those who carry the mutation, but the hope is that these treatments will delay or even prevent the disease occurrence, because research is showing that this LRRK2 protein is overactive in people with PD, whether they have this gene mutation or not. And so the hope is that they'll study these drugs in people with a mutation, but then the treatment will be applicable and more far-reaching than to just those who harbor that mutation. Yes.
Question, if I can jump in.
Please.
Specifically, like with Denali, what is it that they're, I know they're looking to target the LRRK2 in going directly to those problem proteins. What is it that medication that's in the pipeline, what will that help? How will that help a person with Parkinson's?
So I don't actually know. I mean, I don't know the answer to that, because I don't.
Or is it directly toward the movement? Is that what it is, do you think?
Yeah, I don't want to answer that question because I don't know the answer to it. I don't exactly, it's not, the stuff is pretty
Hush hush.
Yeah, so I don't know exactly where in the pathway that this is even targeting. All I know is that it is, trying to make that protein, instead of acting abnormally and be overactive, it's trying to decrease its activity. So my guess would be that it's the movement component, but don't anyone put money on that.
Yeah. Well, the other component you talked about was slowing the progression. Some of the therapeutics are targeted there. Does that mean that those medications might be considered neuroprotective? Is that
yeah?
Fair?
That's the hope. Yeah. So the idea is maybe that having an overactive LRRK2 gene, it has to be overactive for a long time before the Parkinson's actually develops, right? So you have these insults that are happening over a lifetime. And the idea is maybe that if, let's say you identify a person with the LRRK2 mutation, and you can show that they have the overactive protein, that you could go ahead and treat them for the overactive LRRK2 before the Parkinson's develops, and that might actually prevent the Parkinson's from developing. We are a long way from that.
Yeah, I do know, Elaine. I mean, we know that at some point in time, by the time somebody is diagnosed with Parkinson's, they've lost a pretty substantial amount of their dopamine.
Yeah, so they've lost, 60 to 80% of their dopaminergic neurons by the time that they're diagnosed. So this thing has been set in motion long before the symptoms come on.
I was just wondering if they've corresponded LRRK2, where it's, how it progresses, its length of time expressing itself to the time that people, seems like it almost would correspond.
Yeah, and I don't know the answer to that. Maybe they have some of that data in animal models, probably not in humans because we can't really look at, well, with better imaging techniques maybe. But
I
yeah,
don't
I
know.
mean, that's the
Thought
route
we
where
had
they're
the
going.
key.
That's where they want to be.
Thought we were going to unlock it. I'm going to shut up now. I'm going to get off.
No, you're good. All right, so the other gene that clinical trials are underway with are for the GBA gene. And the Silverstein Foundation for persons with Parkinson's with GBA actually launched a company called Prevail Therapeutics, and they raised $129 million towards investigating gene therapies for this particular gene. And this is Prevail Therapeutics here. And they have begun clinical trials recently. And the first drug to be evaluated was Ambroxol. This was actually one of those drugs that Vince kind of brought up where they were able to repurpose an old drug for a new therapy. And so the next slide, we're going to go through how that happened. And this one's in clinical trials, but we don't have data back yet on the trials in people with Parkinson's. But the hope is that this drug is going to improve the function of this protein in people with this gene mutation. And third company that was launched in 2017, Genzyme, also has a clinical trial looking at another drug that also is targeting a fatty acid that builds up due to GBA mutations. And this particular study has already enrolled participants. I think they have 243 so far, and they're expected to share their results as well by the end of this year. So there really are a handful of really exciting things coming out, and hopefully we'll learn more about those when we see the data from the clinical trials. So what is in the pipeline? So I want to highlight down here. So if you look down here, you kind of see the pathway of what it takes to go from drug discovery, drug research, to getting it into animal studies and then into clinical trials. It's a long road. There's a lot of money that's involved. There's a lot of time. We're looking at a minimum of 10 years, up to 20 years for this to happen. And so that kind of seems maybe overbearing or not very exciting, kind of depressing a little bit. But if you think about the fact that we've been studying the genetics of Parkinson's disease since 1997, we've actually got, we're quite a long ways in this pathway. And I just wanted to highlight here, these are all novel candidates for new therapeutics based off what the genetics has taught us about Parkinson's disease. And while some of them are just in the beginning of the exploratory research part, many of them have started to work their way up towards this phase 1, 2, and 3 clinical trials, and we're getting really close to having some that will hopefully be approved. And this is going to be a big deal. It's going to be new medications on the market that we haven't previously had access to in the past. So keep your eyes open for exciting things to come. So now let's talk about Ambroxol.
Can I ask a question there?
Yes, please.
You'd mentioned it's new medication, things that we haven't seen before. It seems like what's been coming down the pipeline for the past 3 to 5 years are same medication, just different delivery. So is it going to actually be something that's doing something different, not just something that's delivered differently?
Yes, I can answer that question definitely. Yes, it will be new ways to treat Parkinson's disease.
I'd love to hear that. Thank you.
Yeah, that's exciting stuff. So this gets back to one of the questions that you asked, Vince, is kind of identifying drugs that are used to treat other disorders that maybe now that we're learning more about the biology of Parkinson's disease, we might be able to use them to treat Parkinson's as well. So genetic variations in the GBA gene, we know that those are associated with Parkinson's disease, and that association was first described in 2004. And I have a timeline on the next slide, so we'll dive in more. But we know that from just research studies from basic laboratory science that Ambroxol is able to elevate levels of an enzyme that's called GCase. And this is the enzyme that gets mutated and doesn't function in people who have mutations with the GBA gene. And this enzyme, GCase, helps to get waste out of the cell. And Ambroxol is a clinically available drug that laboratory research has shown elevates this protein, but it's also currently approved and in use for patients with a respiratory condition. So the idea is that maybe we can actually repurpose this drug for Parkinson's disease. And so this is probably going to be the first one that comes out pretty soon. So let's talk about a little bit about what that looked like. And I call it going from the bench to the bedside, right? So we're taking the science that happens in the lab on a bench and moving it into real clinical practice. So in 2004, genetic variants of the GABA gene were identified and associated with Parkinson's disease. They are actually some of the most common genetic risk factors that we know of. And it's believed that about 5 to 8% of Parkinson's patients have one of these mutations. So Ambroxol was identified as a potential candidate, and preclinical research was done in mice and primates, and it showed that Ambroxol can actually increase the levels of this enzyme that's disrupted. These studies also showed that Ambroxol treatment could reduce the amount of Parkinson's associated protein alpha-synuclein. So again, we're seeing some overlap here, right, in what we see by studying these different genetic events. And so based on these results, the researchers were able to justify and get approved a clinical trial, and to start with just testing the safety of the drug in people with Parkinson's disease. And so they did that, and these first go at these clinical assessments suggested that this treatment did improve the motor features that were associated with Parkinson's disease. And to quantify that, they had a 6-point reduction in their UPDRS-3 scores. And then what's really interesting and exciting is that these effects were also observed in patients, participants who have Parkinson's disease but do not have the GBA mutation. So this is a pretty exciting finding that we are now able to apply this new drug not just to people with the gene mutation, but also other people with Parkinson's disease. And that gets at, there must be similar biology underlying all of Parkinson's disease. So, you know, I do want to highlight that, so they're still doing the next phase of these clinical trials. And so while the results I've kind of shown you guys sound great and you're like, oh, you know, sign me up, I do want to point out that so these patients who were in this first study, they actually had to take 21 pills of Ambroxol to get high enough dosage, and that's a lot. So that's on top of what they were already taking. And there are efforts now to try and reformulate this drug so that it's not, you know, such a heavy pill burden. So I just want to highlight that while this stuff can sound exciting, there's also risk associated with it, and they didn't know that these results were going to turn out great. So there are many considerations that one should think of before, you know, signing up for a clinical trial. It can be a great thing to do, but there are considerations for that. So let's talk about this testing. So I don't know how many people on this call have undergone genetic testing, have thought about genetic testing, or even know where to go to get genetic testing. But like we said earlier, you know, it's not something that's commonly been done from a treatment perspective. So I want to share a little bit about the story of Genia Bryn. So, you know, her testimonial is that 15 years ago, she was diagnosed with Parkinson's disease, and this diagnosis brought back childhood memories of her aunt who also had Parkinson's disease. And so for her, this was the first time that she considered this disease might run in her family. And so one of the things that we should all think of before we go and get genetic testing is that this can not only influence us, but it can have an impact on our families as well, right? Because especially if you have children. So, I mean, if you carry the gene, it means that your children could have a 25% chance, 50% chance, whatever that might be, of also carrying that gene, and maybe they don't want to know, or maybe they do want to know. In her case, her husband and both of her sons decided together as a family to find out if they had the mutation or not, and they did. it turned out they had a mutation in the LRRK2 gene. So it was a family decision, and I think this just highlights the carefulness that should be associated with making a decision like this. And I really like her son's perspective. So many of you have probably heard of Sergey Brin, who is her son. He's also the co-founder of Google. But what he says about this is that this leaves me in a rather unique position. I know early in life something I'm substantially predisposed to. Now, mind you, it's 20 to 80% risk factor, so it goes from really low to high, and who knows where it is in that spectrum. But his perspective is I now have the opportunity to adjust my life to reduce those odds. I also have the opportunity to perform and support research into this disease long before it affects me. And he goes on to say, that all of us are going to have some kind of condition in our old age, if we're lucky enough, right, to live that long. And he has a better guess of what his ills may be. And I think that's a really great way to look at it. Doesn't mean he's going to get it, but it does mean that he can go ahead and make lifestyle choices that might protect him from it, or maybe just delay the onset of it.
Alaine, I have a question.
Yes, please.
How accurate are those 23andMe DNA tests?
Very good question. So they are accurate. If the mutation is known. So here's the thing, right? Like if they sequence the gene and they're looking for a specific mutation that's described, either you have it or you don't. They can explain that. But maybe you have a novel mutation that hasn't been discovered yet. They're probably not going to pick that up. But they're accurate. I mean, they are accurate.
Thank you.
Yes, you're welcome. Thank you for the question. And then I just want to point out, this is Anne Wojcicki next to him, and she's actually the founder of 23andMe. And so between the two of them, they've actually donated over $100 million to Parkinson's research. And I actually remember back when I was in grad school at Emory, 23andMe just started, and it was really exciting to get on their website and follow them as they began to identify more and more mutations that were associated with Parkinson's disease. And this was somewhere in the early 2000s. So right after alpha-synuclein had been described and a lot of these things started to come out. All right. So Michael J. Fox Foundation did a survey on genetic testing, and they found that most people had never been tested or were not planning to be tested. And so I just wanted to share a little bit of information at the end here with, if you are interested in being tested or you want to talk to somebody more about it, some places that you can go. So of course, whoever your physician is that you mostly see for managing your Parkinson's disease would be a great place to start. Fox Insight is a collaboration with 23andMe, and they will do free testing for people with Parkinson's disease, and they look at the GBA and the LRRK2 genes. PD Generation is a collaboration with the National Parkinson's Association. It's also free, and it also offers free genetic counseling, and it looks at GBA, PARK7, SNCA, LRRK2, Parkin, and PINK. And you have to have a diagnosis of Parkinson's disease to get these free genetic testing services. And then more locally, I know that at the Mayo Clinic in Jacksonville, UF Shands in Gainesville, University of Florida in Jacksonville, as well as some of the local movement disorder neurologist offices in Daytona do genetic testing. And so those could be good places to start as well. And that is all I have. So I'll open it up if you guys have any more questions, or if you want to put those in the chat box, or if you want to just email them to Vince. My email's here as well if you have questions that you might think of down the road.
There is a question, actually a couple of questions.
Okay, great.
One of our friend John Zells, he was a little late in getting to the program today, but he wanted to remind you about a question that was posted on Facebook support group. If you haven't already addressed it, and I don't believe you have, but maybe you have, but can you please discuss the genetic risk of passing along the SNCA2 gene mutation?
Yes, thank you for reminding me about that, John. And I'm going to go back to this slide here because there's not a specific answer to this. And it is because of the fact that it depends on which mutation you have in that particular gene. So I know that SNCA2 gene is associated with Parkinson's disease, but also different types of epilepsy, among other things. So it really depends on which mutation you have in that gene and how that inheritance pattern is. I believe SCNA is a autosomal dominant inheritance pattern, but there are many different mutations associated with that particular gene. And so, unfortunately, I cannot answer that question, but I can say that there's probably a 50% chance if a parent has it that they are going to pass it along to the child. And then again, it's like these other ones, it might not be that having that mutation means you're going to have the disease. It means that you could have an increased risk of the disease. And so that's probably somewhere, and I'm just guessing here, on the order of 2 to 15%. But then it's also going to interact with your environment, with your lifestyle choices. And so there's not a specific answer to that.
No, we do have another question.
Okay.
From Lee Wygand, I hope I pronounced that last name correctly, states that I've undergone genetic testing from 23andMe, and I tested negative for both LRRK2 and GBA genes. So maybe some other gene variation has contributed to my Parkinson's?
It's a good question, Lee. It's possible. So one thing to remember is that only about 15% of people with Parkinson's have a genetic cause. So the chances of you having a genetic form of Parkinson's disease is pretty low, but definitely, and if you have a family history, so like if one of your parents have it, an aunt has it, uncle, grandparents, then it is more likely that that it is a genetic form. And yeah, there, like you see on this slide here, there's to date 23 different regions identified, and 23andMe only test for 2 of them. So yeah, it's likely that there's some other variation.
This is from Sheila McGee. She asked if you don't have Parkinson's disease, but your parent does, where can you get tested? I think you had a slide on that.
To get tested by Fox Insight or PD Generation, from what I can tell on the website, you personally have to have a Parkinson's diagnosis. However, if you see a movement disorder neurologist or even a primary care physician, they can order these genetic tests from different companies, and they're not that expensive. So I would, if it's something that you're interested in, just talk to your physician, and they can definitely get you to get you tested.
Kathleen Williams is asking if will you be providing us with a copy of the presentation, the slides?
Yeah, I'm happy to do that.
And just a comment from Gail Balin, who I think it is. She's a speech language pathologist. She just comments, excellent presentation.
Thank you, Gail.
Yes, and that's and thank you. We're coming across the board. No other comments at this time.
Thank you, everyone, for joining us today and helping us keep connected.
Yeah, well, thank you, Alaine. Really excellent stuff. I mean, we're truly grateful for all this information, and thank everybody for attending today's program. And we hope to see you next time.